The topic of today's lecture is the major cellular catabolic process called citric acid cycle. Uh, this is chapter 16 of Langer textbook, but before talking about specifics of this topic, let's recap another catabolic process which we already talked about, the glycolytic pathway. Once cells consume glucose, it can be drained down to the glycolytic pathway to bring it down into smaller tricarbonic pieces, which are called pyruvate. In anaerobic conditions, as we discussed, the reducing equivalents which are generated from glucose during the pyruvate productions are turned back on pyruvate to reduce it to either lactate or ethanol and CO2, which are respectively called the lactate or ethanol fermentation process. And these are the processes that cells can use to produce energy from glucose in anaerobic conditions. And as we know, the fermentative glycolysis is extremely inefficient process to produce energy. Only 2 ATP molecules can be produced per glucose as a result of fermentative glycolysis. The reason for this inefficiency, it's very easy to understand if we consider the energetics of the process. In fermentative glycolysis, so the energy release from glucose beginning of the pathway to pyruvate or lactate and ethanol, which are not shown here, but the energy release is very small, approximately 100-200 kilojoules per mole. And again, this could be understood because at the end, the end product of the anaerobic fermentative glycolysis is some bond rearrangement within the glucose molecule without net oxidation or reduction. But if you would imagine a process that would faithfully burn glucose by atmospheric oxygen into CO2 and water, this process could potentially release approximately 20-fold more energy as compared to fermentative glycolysis. And cells evolved such process during the rise of oxygen-rich atmosphere, which is collectively called cellular respiration. Strictly speaking, cellular respiration is a set of catabolic processes where cells produce catabolize different kinds of organic matter into CO2 and water by consuming atmospheric oxygen. And this process, as we will see later, is much more efficient in terms of energy production as compared to glycolysis. So let's look closer at the this catabolic process, set of catabolic process, cellular respiration, and the place of the citric acid cycle in this framework. The cellular respiration can be brought into 3 stages. First stage is called production of acetyl-CoA. Second stage is oxidation of acetyl-CoA, and third stage is oxidative phosphorylation. The purpose of the first stage is to convert all different kinds of organic matter catabolized into uniform active acetyl pieces in the form of compound called acetyl-CoA. Acetyl-CoA is an acetyl group which is bound to a specific carrier called coenzyme A, which we will discuss later. During this process, although some ATP could be generated, some reducing equivalents could be generated, and as in the case of glycolysis, some CO2 already can be produced, but the most important contribution of stage 1 is production of acetyl-CoA, this uniform 2-carbon pieces that could be fed into stage 2. Stage 2 is an oxidation of acetyl-CoA. In this stage, these uniform 2-carbon pieces are completely grinded, oxidized into carbon dioxide and water, concomitantly producing reducing equivalence in terms of reduced form of various electron transport carrier. So this is a set of catabolic reactions. It consists of 8 steps and starts with production of citrate. And because of this, it's a roundabout cyclic nature, and the fact that the first intermediate is citrate, it is called citric acid cycle. So at the end of citric acid cycle, the end products, the reduced electron transport carriers, are fed into electron transport chain that uses a set of protein complexes sitting within inner mitochondrial membrane to transfer gradually these electrons to atmospheric oxygen, thus converting the reduced form back into oxidized form and water. And the power of this electron transfer is used to fuel or to power the proton pumps that generate electrochemical gradients, which is used then for ATP production. So this way, if one would take an analogy of car engine, so the first 2 stages could be sort of as fuel preparation steps, and the third stage would be could be compared to burning the fuel in the actual engine and cylinders to produce the power. So altogether, the citric acid cycle is a major intermediate step in the cellular respiration, where during the set of roundabout of catabolic chemical reactions, the acetyl groups fed into citric acid cycle are converted into CO2 and reduced form of electron transport carriers. The initial step of this catabolic pathway is production of citrate, which gives the name of this cycle of chemical reaction, citric acid cycle. But besides the role of citric acid cycle as a major catabolic process, it also has an anabolic role. The stages of different intermediates in the citric acid cycle, as we will see, are also important branch points for production of other biomolecules. For example, first step, citrate, is measure step, also measure intermediates in production of various kinds of lipids. Some other intermediates such as alpha-ketoglutarate or oxaloacetate are important precursors for amino acids and the nucleotide bases. Another intermediates such as succinyl-CoA are important precursors for polyunsaturated compounds such as porphyrins, which are used in enzymes and other protein prosthetic groups. So as such, the citric acid cycle is said to be amphibolic pathway, it having characteristics of both anabolic and catabolic pathways. The topics of today's lectures are several fold. First, we'll look at chemical logics of first 2 steps of cellular respiration, production of acetyl-CoA, and the stages of the citric acid cycle, specifically focusing on the starting point of this process, starting with the endpoint of glycolytic pathway pyruvate. and then we will look at how these processes are regulated from the standpoint of the enzymes participating in citric acid cycle and from the point of intermediates which are used to fuel citric acid cycle. And then we look at amphibolic nature of citric acid cycle, specifically focusing on its anabolic functions. To best understand today's lecture, please take a look at Fergus lectures on enzyme reactions and bioenergetics and my previous lecture on glycolysis.